Insights, Basics and Beyond, Chapter 65, Shinya Yamanaka and Induced Pluripotent Stem Cells. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our recent chapters, we explored how immunotherapy can be used beyond cancer to calm autoimmune diseases, support the fight against infections, and even reshape allergic responses. All of those strategies depend on the cells and the molecules the immune system already gives us. Today, we take a small step back from specific diseases and focus on something more fundamental: how we can rebuild cells themselves. Our topic is Shinya Yamanaka and his discovery of induced pluripotent stem cells, or iPSCs, a breakthrough that changed regenerative medicine and opened up new possibilities for future immune-based therapies. So let's talk science. Who is Shinya Yamanaka? Shinya Yamanaka was born in Osaka, Japan in 1962. He initially trained as an orthopedic surgeon, but early in his career he realized he was more drawn to understanding diseases at the cellular and molecular level than to the operating room itself. That curiosity eventually led him into basic research in pharmacology and developmental biology. He worked in several research environments in Japan and the United States before returning to Kyoto University, where he began asking a deceptively simple question: is it possible to take a fully differentiated adult cell and rewind it back into a stem cell? At the time, the dogma was that cell differentiation was essentially a one-way street. Yamanaka's work would turn that idea upside down. So let's clarify first, what are induced pluripotent stem cells? Before iPSCs, there were two main stem cell types in the spotlight. First, embryonic stem cells, or ESCs, derived from early embryos and capable of becoming virtually any cell type, so being pluripotent, but ethically and practically controversial. And second, adult stem cells, tissue-specific progenitors, for example like hematopoietic stem cells in bone marrow, with more limited differentiation potential. Yamanaka's idea was radical. Instead of starting from embryos, could we reprogram adult somatic cells back into a pluripotent state simply by changing gene expression? Working with mouse fibroblasts, his lab screened combinations of transcription factors, genes known to be important in early development. After testing many combinations, they found that a set of 4 factors was sufficient to reprogram differentiated cells back into pluripotent ESC-like states: OCT4, SOX2, KLF4, and c-MYC. Together, these became known as the Yamanaka factors. Cells reprogrammed with these factors behaved like embryonic stem cells. They could self-renew and differentiate into many different cell types under the right conditions. Soon after, the same strategy was applied to human cells, creating human iPSCs. In 2012, Yamanaka shared the Nobel Prize in Physiology or Medicine for this discovery. So, how did iPSCs change the game? Yamanaka's discovery had several immediate consequences. First, iPSCs were an ethical and practical alternative to ESCs. iPSCs offer embryonic-like pluripotency without the need to use embryos. This removed a major ethical barrier and greatly expanded the practical possibilities for stem cell research. Second, they were a patient-specific cell source. Because iPSCs can be generated from a patient's own cells, for example skin fibroblasts or blood cells, they make it possible to model disease in a dish using patient-derived neurons, cardiomyocytes or immune cells, and also potentially generate autologous cell therapies with reduced risk of immune rejection. And third, they can be applied for regenerative medicine and tissue engineering. iPSC-derived cells are being explored for retinal pigment epithelial cells for macular degeneration, cardiomyocytes for heart repair, and dopaminergic neurons for Parkinson's disease. For immunology, this means we can imagine making new immune systems, or parts of them, starting from reprogrammed adult cells. So let's look now at the connection of iPSCs with the immune system. Where do iPSCs intersect directly with immunology and immunotherapy? First, for disease modeling in immune-mediated disorders. From patients with primary immunodeficiencies or autoimmune predispositions, researchers can derive iPSCs and then differentiate them into relevant immune cells, for example T cells, B cells, or macrophages. This allows then a detailed study of disease mechanisms in human cells and also testing of drugs or gene editing strategies on a patient-specific platform. Second, they help manufacturing future immune cell therapies. In principle, iPSCs can be differentiated into T cells or NK cells for adoptive cell therapies, but also dendritic cells for vaccines, or into universal immune effector cells engineered for off-the-shelf use. This could, over time, complement or even partly replace the laborious process of harvesting and expanding cells from each individual patient. Third, they paved the way towards universal donor lines. Another concept is to create HLA-edited iPSC lines with low immunogenicity, so universal donor cells that can be banked and used across many patients with minimal rejection risk. These iPSC-derived cells could become a standardized starting material for many cellular immunotherapies. And fourth, they support testing gene editing and combination strategies. iPSCs provide a flexible platform to combine CRISPR-Cas9 gene editing, synthetic receptors, for example the CARs, and safety switches or logic-gated circuits. Once validated in vitro, edited iPSCs-derived immune cells could be brought closer to clinical application. Of course, important challenges remain, such as the risk of genomic instability, potential tumorigenicity, and ensuring fully functional mature immune cells, but the conceptual platform is enormously powerful. Now let's look at a more personal episode in Yamanaka's life, or how he went from clumsy surgeon to Nobel laureate. One of the more charming stories about Yamanaka comes from his early surgical career. As a young orthopedic surgeon, he was known for being meticulous but slow in the operating room. After one particularly long procedure, a senior colleague reportedly remarked that if every case took him that long, the hospital would soon be out of business. Yamanaka has also mentioned that his wife, herself a physician, once watched him operating and later told him gently but honestly that he might be better suited to research than to surgery. He took this seriously, shifted his focus toward basic science, and eventually chose the lab bench over the scalpel. The result of that decision? A discovery that fundamentally reshaped stem cell biology and opened new horizons in regenerative medicine and immunotherapy. It's a nice reminder that not being brilliant in one thing can open the door to being transformative in another. When it comes to iPSCs, of course there are limitations and open questions. Despite the excitement, iPSCs are not magic bullets. Challenges include: first, genomic and epigenetic stability. Reprogramming and prolonged culture can introduce mutations or epigenetic abnormalities. For clinical applications, iPSC lines must undergo rigorous quality control. Second, maturation and function. iPSC-derived immune cells sometimes behave more like fetal or immature cells than like fully mature effector cells found in adults. Achieving functionally robust, long-lived immune cells is still an active area of research. And third, safety and standardization. Any residual undifferentiated iPSCs in a therapeutic product could potentially form tumors. Protocols must ensure robust differentiation and include safety measures before clinical use. However, even with these challenges, Yamanaka's work has fundamentally expanded our toolkit. We now know that cell identity is not fixed, but programmable. To conclude, Shinya Yamanaka's discovery of induced pluripotent stem cells turned the idea of fixed cell fate on its head. For immunology and immunotherapy, iPSCs mean new ways to model immune and inflammatory diseases in patient-derived cells, potential sources for standardized or personalized immune cell therapies from T cells and NK cells to dendritic cells and beyond, and a conceptual foundation for integrating gene editing, synthetic receptors, and regenerative medicine into a unified programmable cell therapy platform. In the last few episodes, we have seen how the immune system can be turned to fight cancer, calm autoimmunity, control infections, and mitigate allergies. With Yamanaka's work, we now add another dimension, the ability to rebuild and redesign the cells themselves. So, this is a perfect bridge into our next block, where we will talk about future directions in immunotherapy, starting with personalized medicine, how genomics, biomarkers, and data driven profiling are helping us choose the right immune therapy for the right patients at the right time. Before closing, I have a small request. If you enjoyed my content and want to hear more, please like and subscribe. It's a small step for you, but it helps me a lot. So, thank you for listening and see you next time.